The overall goal of the following experiment is to investigate the spatial organization of specific genomic loci in single cells in a robust fashion. This is achieved by first generating fluorescently labeled probes to ultimately bind and highlight the genomic region of interest. The next steps are to adhere, fix, and permeabilize cells of interest on a slide in order to permit probe access. Then a simultaneous heat denaturation of probe and cellular DNA is followed by overnight probe hybridization, wash steps to remove nonspecifically bound probe, and counterstaining with DAPI to visualize the cell nucleus. Results obtained with fluorescence microscopy will ultimately reveal the nuclear position occupied by specific genomic regions.
This method can help answer key questions in the nuclear organization field, such as determining functional interactions between genes.
The main advantage of this particular DNA FISH protocol is that it is fast, robust, and versatile.
Although we demonstrate this method using ES cells, it can also be applied to numerous other cell types.
Directly labeled probes made from BACs of 100 to 250 kilobases consistently produce bright signals. If smaller probes are required, use 40 to 50 kilobase phosmids or even plasmids containing 5 to 10 kilobase inserts. Identify the BAC or phosmid clones corresponding to specific genes using the Ensemble or UCSC genome browsers. To prepare high-quality BAC DNA, use either repeated precipitation or a commercially available kit. The cleanliness of the preparation is proportional to the volume of probe required.